Are we live? Okay. Hello, everyone, and welcome to the online. Oh, wait a minute. Is there? I don't have any video. Can you?
Is the size of the window okay, or should I make it smaller or larger?
No, it's okay like this. Yeah.
It's okay.
So it's just I realized that my camera is off.
It says we are live for 27 minutes.
Yeah. Okay,
seconds.
Yeah. So we had a bit of a technical problem now. But hello, everyone, and welcome to the online version of the Neuro Forum, organized by Tim Vogels at the IST Austria and Andrew Sachs at the University of Oxford, UK. I'm Everton Agnes. The camera is not working now, but I'm here, and I'm today's talk host. I'm a project leader at the Biozentrum, the University of Basel in Switzerland, and I was a postdoc with Tim Vogels until the end of September. So before introducing today's speaker, I would like to mention a few things. First, you might be expecting Tim Vogels or Andrew Sachs as hosts of Neuro Forum. As usual, they are the hosts. So a short explanation is that months ago, while I was still in Tim Vogels' lab, a new schedule of speakers was organized and also decided that the postdocs and PhD students would be hosting that, and that's why I'm here today. Second, I would like to ask you to use the chat on your right as much as you want. And if you want, or if you have any questions that should be asked during the talk, please write them there in the chat and I will ask them during the talk. Third, if you have questions that you like addressed at the end of the talk during the Q&A, please use the questions box at the bottom. There is an ask a question. Please mention there whether you like to be invited on the screen to ask the question or whether you like me to ask the question. So now to the talk. It's a pleasure to have Professor Tatiana Chumachenko with us today to present the latest work from her lab. Tatiana has just been appointed as W2 Professor for Computational Neuroscience of Behavior at Bonn University in Germany. She's also an independent research group leader at the Max Planck Institute for Brain Research in Frankfurt, Germany, where she started her own group in 2013. Before that, Tatiana did a postdoc at the Center for Theoretical Neuroscience at Columbia University, New York, and a postdoc that was joint appointment at the Max Planck Institute for Dynamics and Self-Organization, the Bernstein Center Göttingen, and the Interdisciplinary Collaborative Research Center in Germany. Tatiana did her PhD in computational neuroscience at the Göttingen University in Germany, and her undergrad or diploma in physics at the Technical University of Darmstadt, also in Germany. Amongst many awards, Tatiana has just been awarded a prestigious ERC starting grant from the European Research Council. So without further ado, I would like to invite Tatiana. Oh, so she's already sharing the screen. So I would like to invite Tatiana to present her work on the emergence of contrast invariance in cortical circuits.
Thank you, Everton, for the very nice introduction. It's a pleasure for me to present my work here. So when I say my work, it's actually the project that has been led by a talented PhD student in the lab, Laura Berner-Estimot. So the project is about the emergence of contrast invariance in cortical circuits. So if we were, all of us, if we were at IST right now, which is the host institution for this particular talk, what would be seeing in the morning is this picture of IST, basically with bright lit colors, where we have no trouble recognizing the scenery around us and to recognize the houses and the lake. So the objects that appear in the morning in bright contrast are actually the same objects that are there in the evening, and we are still able to recognize those objects, even so they are presented now at a lower contrast. And that is the feature that I want to talk to you today is the question how we are able to recognize visual stimuli across different contrast levels. And that particular feature is called contrast invariance. It means that the invariant object recognition is present regardless at which contrast that object is presented. In that particular talk, I would be focusing on the visual system, and therefore the input is the visual input. The typical presentation of an input in the model would be differently oriented bars. And the contrast is really the difference in brightness from one presentation of the object to the next. Let me move on to explain, and this is something that many of you potentially are familiar with, but I would repeat this very briefly for the new students who are listening to the talk. The hypothesis evolving contrast invariance is this, that the contrast invariance that we experience on the behavioral level is rooted in the activity of the visual cortex, meaning that there's something about the activity of the visual cortex that contributes to the perceptual invariance across contrasts. And there has been this historic finding in the visual cortex, first reported by Hubel and Wiesel in the 60s, meaning that if you record from different neurons in the visual cortex, what you would find is that they are selective for a particular object, and that is a bar which is oriented in a particular way. And if you vary the orientation of this bar, you will find that the response of this neuron, here the number of spikes as a function of second, in response to differently oriented bars would vary, and there is a object orientation that is triggering that neuron best, meaning that the neuron has the highest number of spikes emitted, and then there is a Gaussian, almost Gaussian fall-off to the left and to the right of the preferred orientation. Now, if you want to characterize what the response of the neuron is, or of the population of neurons for that matter, what people look at is the tuning width. This is the full width at half maximum across this particular Gaussian. What happens if we look at different contrasts? There are obviously contrasts that are high or contrasts that are low. What happens to the tuning curve of these neurons? Does it get changed in some complex nonlinear way? Or is it changed in a way that we can relate to the contrast that that particular stimulus was presented at? And it turns out that if you plot the tuning curves of these neurons that have been recorded at different contrasts, what you see is a picture like that. These are simulated data. What you see at contrast, that is intermediate contrast strength, you would find a peak amplitude which is lower, obviously, so all of the fine rates are lower for this lower contrast value. And if you increase contrast to a higher value, you would find that the whole curve is increased, so there is a higher peak response rate. However, when you rescale the tuning curves with respect to their peak height, what you would find is that there is actually an invariant curve emerging, and that is the mathematical definition of contrast invariance. Again, contrast invariance means that the shape, and in particular the width of the tuning curve, is preserved across contrast. And we believe this is the substrate generating the invariant perceptual, the invariant percept of objects across different contrasts or different luminosity levels. And by the way, guys, if you have any questions, please feel free to interrupt and ask me anytime. So you don't need to wait until the end of the talk to ask any question. So let me move on to nail down the location of contrast invariance in the visual system. The contrast invariance is present at two levels in the visual cortex. It is present in the individual neurons, meaning the tuning curves of individual neurons can be contrast invariant, or it can be present at the level of populations or whole orientation maps. And these are two citations associated with the two findings. So at the level of individual neurons, what I mean here is that if you record these curves at individual neurons, neuron 1 or neuron 2, and you do the procedure per neuron, you can find, and this is again the Anderson et al. 2000 citation, that the tuning width at low contrast and the tuning width at high contrast are actually nicely correlated and lie along the diagonal with a very high R-square value. And similar things happen on the orientation map level, meaning that Carandini and Sengpil 2004 have shown that the orientation structure, the structure of the orientation map is preserved regardless whether the structure was measured at low contrast or high contrast. suggesting that there is a contrast invariant feature of orientation maps that is present in the CAT-V1 cortex. Now, the question is really the question here is really, is the contrast invariance of individual neurons the thing that is generating the contrast invariance at the network level, or are the two somehow separate? That if you have single neurons being contrast invariant, is it really sufficient to generate contrast invariant on the population level? And ask differently, if you have contrast invariance at the network level, is it necessary that the individual neurons are contrast invariant? Or are there ways to obtain contrast invariance at the network level without having contrast invariance in the individual neurons? So the question is it mechanistically enough, or let's investigate the two arrows going from single neurons to population and populations to single neurons. So is it such that the contrast invariance of individual neurons would be enough to explain invariance of orientation maps? It turns out that if you study the balance limit of neurons, or of the populations of neurons, you would find that when excitation and inhibition balance and you achieve the balanced state limit, that the properties that emerge at the limit of perfectly balanced excitation and inhibition inhibitory currents, that this is not sufficient to explain the behavior of the population with respect to the way it responds to inputs. What do I mean by that? In this balanced state limit, the single neuron properties do not determine population activity, because in the theoretical limit of the balanced state network, the response of the network to external stimuli is determined by linear response, meaning that the single neuron properties are washed out at the network limit, and what stays is a linearity that is emerging from the correlation between the inhibitory and excitatory currents in the individual neurons. So the properties that you see at the network level are not inherited from the nonlinearity, so the particular behavior of the individual neurons. They come about through the coordination of excitatory and inhibitory currents. So when you have that correlation between excitatory and inhibitory currents, you cannot say that any population-wide contrast invariance is due to the individual neurons being contrast invariant. So now let's look at the balanced state limit. In that limit, connectivity and synaptic plasticity control the activity of the balanced networks. What do I mean by that? There is nice work by Rosenbaum and Doiron in 2004 showing that the tuning curve of a balanced network is dependent on the synaptic profile of that network and on the input profile delivered to this network, meaning that there is a mathematical theory, the so-called Fokker-Planck equations associated with the balanced state limit, which you can compute if you know enough about the anatomy of the network. And that is a way to predict the tuning curve at the population level. But here, the tuning curve at the population level is not resembling, or it does not emerge from the tuning curves of the individual neurons. It really emerges from the interactions between the excitatory and inhibitory neurons. And similar here, a study, Sadeh et al. 2015 has shown that the modulation in tuning width as a function of the learning paradigm that the animals were subject to can be modulated by synaptic plasticity. This is a theoretical study showing that a particular synaptic plasticity paradigm can introduce before learning a response like that, and after learning takes place and synaptic plasticity could do its job, after learning not only the peak would be changed, but also the width of the response can be changed. This really indicates that there are synaptic mechanisms underlying the regulation of the contrast invariance or contrast dependence at the population level, not necessarily the single neuron nonlinearities or contrast invariant properties. So again, this is a study that the PhD student in the lab, Laura Bernays-Timon, led. And in her project, she was interested about the role of intracortical connectivity and the synaptic plasticity, and wanted to find out how these two features play together to give rise to contrast invariance of orientation maps using balanced networks. So the network model that Laura considered was working as follows. The first ingredient of this model was a leaky integrating fire neuron. This is the differential equation governing the activity of a single neuron. And obviously then the next step would be to connect each individual neuron with other neurons in the network using a statistical approach, and to define a connectivity matrix governing how exactly excitatory and inhibitory neurons would be wired together. The next ingredient in the model would be the tuned input that derives from LGN to cortex, meaning that the currents that the individual neurons in the network are getting depend on the tuning width, or depend on the tuning of the LGN neurons that supply these currents. So these inputs are tuned. And the second ingredient in this model is the orientation-dependent connectivity. What we considered is motivated by a study Marino et al. 2005 that reported that the neurons that have a particular preferred orientation connect preferentially with neurons with the same preferred orientation, meaning that if you consider one neuron in the 2D space and you put a 2D Gaussian around it, this would be approximating the connectivity pattern of any neuron in the network. And the difference between excitatory and inhibitory neurons would be just the width and the amplitude of the connectivity. Now, the next ingredient in our model was short-term plasticity. This is motivated by the reports by Mangilo et al. 2012, for example, saying experimental reports also preceding the study, indicating that excitatory to excitatory connections can be exhibiting short-term plasticity. And there is evidence that not only excitatory plasticity can be present, but also excitatory to inhibitory short-term plasticity can be present. So we really wondered, how do these four ingredients lead to contrast invariance? When does contrast invariance emerge? And when does it not emerge? How can we make mathematical predictions to say what do we expect to find for a particular combination of parameters in any of these four ingredients? So the approach we took is to compare the results of spiking networks to a particular rate model which mimics the activity of the spiking networks. And the rate model is calculated in the balanced state limit using the four ingredients. Now we come to the results. What did we find? To make the results understandable, what we considered first is a network with orientation-dependent connectivity, but with constant synapses. This would give us intuition about what to expect if the neurons and if the synapses of the neurons didn't have any plasticity. This would be the baseline against which we interpret any finding which we find for short-term plastic synapses. So in the balanced state limit, with constant synapses, we find very predictably linear response, meaning that if we stimulate the network with different contrasts, the response to those contrasts would grow as a function of the contrast intensity. However, when we interpret the peak, this is the peak now plotted as a function of contrast, what we would find in balanced networks with constant synapses is simply linear response. And trivially enough, linear response would lead to contrast invariance. So the first finding we have here is a trivial finding, and that is contrast invariance is present in linear networks. Obviously, if every curve is linear, you would have also the peak being linear, and every point is just rescaled by the amplitude of the contrast.
Tatiana, can you explain shortly how is the input? How do you change the contrast as the input to the network?
Yeah. this is a sketch here. The input as a function of contrast is scaled linearly. So there is a prefactor, which we call C, and we increase that prefactor between its minimal value and its maximal value. And this generates us the different curves here.
Okay, thank you.
Yeah, very important question to understand the next slides. So if there are any other questions, please feel free to interrupt me. Cool. However, the linearity of the balanced state is also a problem to understand this contrast invariance, because even so linear networks are by design contrast invariance, we do not see linear responses at the level of single neurons nor at the level of populations. What happens in experiments when they change the contrast, they see curves like that. This is again a log scale. You see saturation as a function of contrast, and it happens for multiple decades of contrast. So first, this is not a linear line in any way. And second, as a function of contrast, this is Bosse et al. 2009 paper. What you can also see is a sigmoid-like curve, and each individual neuron can have a different sigmoid-like curve, meaning that there is a difference in the tuning, in the response properties of individual neurons. But importantly, even at the population level, they are not summing up to be a linear function. So this is the experimental state of the art. And this is the theoretical state of the art in balanced networks without plasticity. So clearly we need to generate something to introduce an ingredient into the balanced networks that would get rid of this linearity and would generate something that resembles the experiments. So I said that at the beginning that the individual properties of the neurons are unlikely to control contrast invariance at the network level. So what is left there? What is left is to play around with the synapses to see if synaptic nonlinearities in the balanced networks can generate nonlinear responses that we see in experiments. And importantly, not only the nonlinear responses, but also a controlled way to understand when the width of the response curves will stay the same and when will it change. So that is the reason we went for short-term plasticity. We introduced synaptic nonlinearities that are due to short-term plasticity in the balanced networks. So what did we do specifically? What we do is we considered Markram-Zoduk's short-term plasticity model as a modulator of the network activity. So what we considered here are parameter regimes that correspond to facilitation, meaning that as a function of the presynaptic firing rate, the release probability will first go up and then decline. And this increase in synaptic release probability, that is called facilitation. And depression is illustrated here. As the presynaptic firing rate is increased, the release probability and thereby the strength of the synapses is going down. Going down means depression. So we incorporated these two synaptic plasticity mechanisms in our model. And what did we find? First, our eyes were on the response of the population or response of the excitatory firing rate, because those are the neurons that project out of the V1 cortex. And we wanted to understand their response profile as a function of contrast. This would be corresponding to the peak of the tuning curve and how it responds to changes in contrast. So what did we find? As we introduced the synaptic facilitation or depression in the model, what we found is that there are ways to get a saturating behavior. This is the bluish curve here. It first starts increasing and then seems to saturate sublinearly. And on the other hand, if we consider facilitation, we can have a supralinear behavior first and then a saturating behavior later. And there are a whole lot of nonlinear behaviors in between that we can get. And mathematically, we can predict these behaviors by considering the balanced state calculations in incorporating synaptic plasticity. So those curves did not occur here by chance. These were curves that we obtained by by studying the mathematics and be able to predict when we have sublinear behavior, when do we have supralinear behavior, and that's why I could show you here two different types of response profiles. Now, the next thing Laura looked at is the network activity and how it is modulated by the synaptic state. And this is the point where we needed to come back to orientation-dependent connectivity and consider short-term plasticity and orientation-dependent connectivity in one model. And STP is the abbreviation for short-term synaptic plasticity here in the next slide. So after introducing this particular Gaussian connectivity profile and plastic synapses, what we saw is that the tuning width does not stay the same anymore. As we increased contrast, we changed the strength of the synapses. That's because we have short-term plasticity in the synapses. And thereby, the firing rates changed, not only due to the change in the input, but also due to the fact that they depressed or facilitated. In this case, this is facilitation, I think, because the increased contrast generates a shrinkage of the response curve. These are the spiking network simulations, and these are the corresponding rate models. So for facilitating transmission, we observed that the contrast responses decreased in the width, and as predicted by the model, the release probability was increasing in a nonlinear way as a function of the presynaptic firing rate. However, the peak network rate was not following the same trajectory as the release probability. Instead, it increased supralinearly its peak activity as a function of contrast. So these are the curves that we obtained after rescaling for the peak, but you now can imagine that the peak grows supralinearly while the width is decreasing if you increase contrast. And obviously, let me mention that when we put in depressing synapses into the model, we saw the opposite effect. This may maybe seem trivial, but I need to say that the model is able to generate the shrinking of response curves, but it can also generate widening, depending on whether you have facilitating or depressing transmission. Now, we started this investigation knowing that the individual neurons do not determine the outcomes at the level of populations and are likely not responsible for the orientation tuning at the population level. However, we wanted to be inspired by the properties of single neurons. And one of the properties of the single neurons is the fact that their spiking rate as a function of the input, this input here is the mean potential, that is measured as a function of varying input, that this particular activation function is a nonlinear function. And this nonlinear function, which is mathematically described by a power law, can be one ingredient in the generation of contrast invariance at a single neuron level. Studies have shown, and in particular the study by Hansel and van Vreeswijk and Troyer and Miller in 2002, have shown that mathematically power laws are the only function that support contrast invariance at the level of single neurons. And in our own lab, the PhD student in the lab, Pierre Eckelmans, could show that leaky integrating fire neurons, when they are fitted close to threshold, close to the spiking threshold, across different input levels and different variability in the input, they can be very well described by a power law. These are indications showing that power laws can be instrumental for contrast invariance. However, these are power laws that apply to single neurons. Now, we wondered if a similar power law structure can be also applied to the synaptic side. So is a power law transformation a requirement also at the synaptic level to maintain contrast invariance? So what do I mean by that? A network with orientation-dependent connectivity and plasticity that is power law type, and I will show you the equations in a moment, where this particular plasticity is present in the excitatory to excitatory connections and in the excitatory to inhibitory connections. Now, the synaptic strength of these connections would be basically governed. There will be a prefactor that is related to the original strength of the synapse. This would be the J0, which I omitted here for simplicity reasons. And then the strength of the synapse will be modulated by the presynaptic firing rate raised to the power of K. And K is something that would be dependent on the synaptic model of your choice. And in the calculations and in the simulations that Laura did, these particular synaptic profiles had the same K. However, there are obviously versions where you have different Ks. However, mathematically, what we find is that it is important to have the same K to generate contrast invariance. So what did we find specifically? Well, Laura changed the contrast from low contrast to high contrast in the model and studied the peak of the response curves and the shape of the response tuning curve. What she found was that the contrast invariance feature was emerging. Even so, the population firing rate, so the peak of these response curves, grew with contrast in a nonlinear way, meaning that there is a supralinear response of the whole network to contrast. However, due to the combination of a particular synaptic change or at the excitatory to excitatory connections and a particular synaptic change at the excitatory to inhibitory connections, the two kind of canceled each other and generated a contrast invariant response at the network level. So this is important because nonlinear networks can be contrast invariant, and contrast invariant in turn does not mean that a particular nonlinear response is present. We can generate different response functions by controlling the exponent K, but still all of these response functions would be contrast invariant. So the two features, the nonlinear response at the population level, the way the peak is scaling with contrast, and the question whether or not it's contrast invariant, these two are really decoupled. This is what we understood using this particular model. So now to the outlook. In our model, we assumed that there is a particular parameter K, which is the power law describing the synaptic plasticity. However, what is the plausible synaptic K for cortical neurons? This is RET-V1, not CAT-V1, but RET-V1. And in these particular data sets, Laura find a K of 0.3. However, I'm sure there are other data sets that would lead to a different power law. However, regardless of which power law you choose, if it matches a particular synaptic condition, it can lead to contrast invariance. Now, importantly, the contrast invariance and the power law synapses can emerge from different mechanisms. It is also possible to consider variability of release probability across individual synapses such that each individual synapse may not be power law, but together they may be acting in the power law fashion if you study the average synaptic strength of a population as a function of rate. What I mean to say here is that the individual synapses can be different, but as long as they conform on the population level to a power law, their predictions would hold. Now, to conclude what I have discussed with you so far and the study that we have conducted in the PhD project of Laura Bender Estimon is this. Synaptic plasticity is a powerful modulator of the activity in balanced networks. This is a figure to remind you of that the firing rate of the excitatory population can behave in a variety of complex ways, which we now can understand mathematically. And networks with non-plastic synapses can be contrast invariant, but if they are balanced, they would likely represent the stimuli in a linear fashion. And spiked short-term plasticity in the excitatory to excitatory connections can permit nonlinear response representation of stimuli, but it would modify the response selectivity, meaning that the tuning width can change depending on short-term facilitation or short-term depression. But if the plasticity is only in the excitatory connections, the width will change. The question is just, will it get wider or shorter? And that will be governed by their particular choice of short-term plasticity in these connections. Now, power law plasticity in the excitatory to inhibitory and excitatory synapses results in contrast invariance can result in contrast invariance based on our math, and permits the nonlinear representation of stimuli combined with contrast invariance. So you can control using one knob how nonlinear response, whatever contrast nonlinear response you wish to model, and by choosing a particular K and ensuring that these two synaptic types have the same K, you can achieve contrast invariance. Now it's time to acknowledge again the people who have contributed to this work. This is my group, and the project that I have presented has been led by Laura Bene-Estimon. This is Laura. Pierre has contributed findings related to the individual neurons and their power law properties, and Andreas Nold has contributed to the project by helping us simulate the networks. Thank you for your attention. I'm happy to take questions.
Thank you, Tatjana. It was a really nice talk. So let's see. I have a few questions that I can ask in the meantime. So people are a bit shy. I think they're a bit quiet today. So just one first technical question that I was wondering in the beginning. If you are comparing the spiking network with the RAID network, right? It's quite nice that you can explain very well the spiking behavior with the RAID network. And I was wondering, what's the size of the spiking network for this to happen, or if there is any thing, because for small networks of leaking degraded and fire neurons, there's some correlations start to break down this comparison.
Yeah, we also noticed, this is a wonderful question. So in the project by Pierre Eckelmans in the group, we really looked at the behavior of finite networks. But here, for the project that I have presented, we took sufficiently large networks, and depending on the parameters, there is a different requirement for what sufficiently large is. But basically, we considered balanced networks, where when we turned off plasticity, we really wanted to see that linear behavior that is predicted by the balance theory. And after we matched that, we put in plasticity. So the typical size of the networks was on the order of, so it depended on the parameters a little bit and the conductivity of the networks, but I think 50,000, 100,000 is a good estimate.
Okay, so it was fairly big. So yeah, okay. Yeah.
But another project in the lab looks into how contrast invariance can emerge for smaller networks that have 1,000 to 10,000 neurons, where single neurons are nonlinear. And there, Natalia Karnikova here, who is Natalia? Here. Natalia uses the data from Laura Busse's lab. These are the data sets that we have shared, that we have shown here in these slides, to define conditions on synaptic wiring diagram using the observation of contrast invariance in small networks. There are a number of projects in the lab that that can work for the smaller network limit and the large network limit.
Yeah. Okay, thank you. So I have so someone, Richard Lonsdale, wants to ask a question. Let's see if I can invite him to the screen. Let's wait. So he's connecting. So another thing before he enters, it's taking some time for him to connect. Now I have another. So you, from what I understand, you have the short-term plasticity between E to E and E to I, right? So have you thought about using inhibitor to excitatory short-term plasticity, so facilitating synapses, for example, or something like this, or it doesn't change? I don't know, or you tested it?
Laura has conducted balanced network calculations for different types of plasticity, and it can be done. I haven't included those results here, but yes. But
they don't change much, the results, or it's not?
So it is important that you have this particular combination of excitatory to excitatory and excitatory to inhibitory connectivity to have contrast invariance. If you break that, regardless of where you put your plasticity in and what the plasticity details are, you will not get contrast invariance. So if you're fine with this, then you can control the nonlinearity any way you want.
Okay, so the two key ingredients are the E to E and E to I.
Yes.
Yeah. Okay. So I don't know what's happening to Richard. So he's not entering. Let's see. I have more questions now. So Chaitanya is asking if there are any systems where contrast invariance or normalization can occur without network level, perhaps at the sensory receptor level.
Yes. Yes, absolutely. Here in this talk, I wanted to show you a mechanism of how contrast invariance can emerge from the interactions of excitatory and inhibitory neurons. But obviously, there are other mechanisms at the neuron level that would guarantee contrast invariance for individual neurons. And I'm sure there are ways to do that in the sensory neurons too. But this was not the topic of our study. We really wondered about circuit mechanisms.
Yeah. So Richard cannot connect. Let's let me see if he can ask. And let me see if yeah. So it's the first time I'm hosting a talk, so sorry if I'm a bit slow with the questions here. So he's asking if there is feedback from the cortex to the thalamus, which might modulate input to the cortex and thereby affect contrast invariance. So he's asking if you are aware of anybody looking into this, either with modeling or experimentally.
I know that this is an important topic. A number of labs are not only familiar with and are diving into the details of it. There was a concerted effort at the level of multiple groups that I was part of, and the question really tries to zero in on a particular mechanism that emerges from the feedback from the thalamus to cortex and from cortex to thalamus. So whether there is anything that can generate you contrast invariance if cortex fail to do so. The question is, we haven't studied it yet, but I think it would be a wonderful thing to address. You start with one contrast non-invariant cortex and try to generate feedback so that it compensates for it. It can be done, we just haven't done it yet.
Yeah. Cool, thanks. So let's see, I think there is no more questions here. Thank you. So I would like to thank you again, Tatiana. And also, so there is a chat at Zoom now. So whoever wants to enter, the link is on the right. Some team just posted it. I can post it again if I can copy. Yeah, so everyone who wants to chat now, you can click on the link on the right, and we will be there in a few seconds or minutes. And I'd like to thank you again, Tatiana. It was a really nice talk. And really,
thank you for the invitation.
Thank you. So let's go there now. Bye-bye.
Bye-bye. Do I need to do something?
I think I end the broadcast.
Okay.